Hello, my name is Madeline Young. I am a student at the University of Southern Maine, and this research focuses on the growth of smallmouth bass in rivers with a restoring river heron run. It is done in conjunction with Matthew Brewer's work, looking at the Penobscot River restoration project, an assessment of the connectivity between the systems now that that project has been done, and Karen, under the supervision of Dr. Karen Wilson of the University of Southern Maine. The purpose of the research is to determine how the growth rates of top predators, especially smallmouth bass, are affected by access to high quality prey, like the river herring, especially alewife in this area, and the various environmental conditions that may affect how that growth rate changes. To determine the growth, we looked at the otoliths from smallmouth bass, aged whole and then sectioned in order to determine the accuracy of each aging method. And then using ImageJ, we inferred the annual growth rates and back calculated the linked at age. The importance of this is really that river herring are important because they transfer nutrients between the marine and freshwater systems due to the fact that they spend part of their life cycle in each system. The Penobscot River Restoration Project, which removes dams from the river in an effort to restore the river herring runs, was completed in 2016. And the idea is that the bass in the restored river may be eating more river herrings. So we want to ask if the bass growth rates are differing between site and age, or if feeding differs through the ages of the fish. For our methods, the fish that we sampled were collected in collaboration with the University of Maine via boat electrofishing in the lower main stem and upstream tributary sites of the Penobscot. The otoliths were then extracted by dissection and then viewed an image whole under a microscope by two separate readers before reaching a consensus age. Then the otoliths were sectioned through the cross sections and aged again in the same way. The paired otolith ages were analyzed using a precision template to give us metrics such as precision and coefficient of variation. Following aging, the images of the otoliths were analyzed using ImageJ and the application ObjectJ to measure otolith growth rings for the annual growth rates, and it allowed us to also back calculate the linked at age. Finally, the muscle tissue was used for sample analysis of carbon-13 and nitrogen-15, the marine-derived nutrients, and it was the dorsal muscle tissue that was dried and made into a powder that was sent to UC Davis stable isotope facility for the stable isotope analysis. Our results show that the first figure illustrates how when aging the whole otolith versus the sectioned otolith, the whole otolith age was higher than the same otolith aged sectioned. The implications of this are that there is a lot of difference between the aging methods, and of course that the whole ages, which are across the x-axis, and the sectioned ages across the y, that the whole ages were higher than the sectioned. In our research, even though previous research suggested that at younger ages sectioning might not be necessary, we decided to section all the otoliths and use those ages due to the range of fish ages in our samples. The second figure shows the distribution of fish ages according to site. And the biggest takeaway from this is that fish that were 5 years old in the cohort year 2016 were most abundant overall. That's the gray section. And that fish from the cohort years earlier than 2016, meaning they were older than 5 years old, were found at VZ impoundment and tailwater in the Piscataquis. The implications of this are that the restoration project was completed in 2016, the same year that most of the fish were caught or from, and this could be a result of more predatory fish surviving due to the return of the sea run fish, whether that be adding a trophic cascade level or just more access to higher quality prey. The other implication of this is that the main stem where these were found, the older fish. The third graph shows the highest annual growth rates that were at VZ tailwaters in years 10, 11, VZ impoundment at year 8, and the Kennebec site in year 4. This shows that while the trends in this graph are not really strong, there were certain years that there was increases in the growth rates, and the highest annual growth rates were seen in the main stem and in the Kennebec where the river was restored earlier. The final graph shows the marine derived nutrients according to age, and the takeaway from this is that there was no significant trend in carbon-13 with an R value of 0.05 or nitrogen-15 with an R value of 0.078 in relation to the age of fish. This means that the same types of prey are being consumed at all age levels. So that is our research, and thank you for your time.